Stanford University.
Can you hear me like this? Is this better? Okay, great. So, so like Tom said, feel free at any point to sort of interrupt us with questions. Just raise your hand if something is confusing, stuff like that. Also, just like with the other lectures, feel free to email us after class or emails around coursework if you find this that way. So today we're going to provide a little bit of background context, introduce sort of put the endocrine system in place in the context of the other sort of buckets of this class, or I guess sort of think about it generally. We're going to talk specifically then about peptide and steroid hormones, different types of hormones that we're going to see, some specifics, interesting things about them that you might want to know. Then we're going to talk about how the brain controls hormones, so hormone release, and then eventually feeding back to how hormones influence the brain. Sort of this dual communication system that's pretty neat. We're going to touch on all those things today. So Tom is going to start out with some background context.
Okay, so way back when life first arose, things were inside single cells, right? Individuals were single cells who had to interact with their environment. They had to get food, they had to get rid of waste, they had to make sure they were in the right pH and temperature so that they could do well for themselves. And as a single cell, it seems like a simple enough task. But as things got more complicated and multicellular life arose, there arose a really big, important issue for life, which is how cells talk to each other. Cellular communication, okay? And you don't have to worry too much about like the jargony words like paracrine, but it's kind of worth thinking about. There are 4 main ways that cells can talk to each other. The first one being cell-cell contact, where you actually have one cell physically touching another cell, and that seems pretty understandable. It's got to be really, really short range, and it's just going to be this cell talking to another cell. We've got paracrine, which is a little more short range, and you guys have heard about neuronal and endocrine. And I'm going to try to put these examples of communication in the context of us as a giant multicellular organism, this class right now, okay? So who wants to be the pancreas? No one wants to be the pancreas. Okay, we got a pancreas right there. Thank you very much. No, we don't have to be specialized cells, but the point is that a cell-cell contact in our organism would be actually someone physically handing you a note during class, okay? You actually, and it's just one-to-one, short and easy. Not a lot of people are gonna know about that signal. Paracrine is more like kind of you whispering to a bunch of neighbors, right? A couple people can hear you. It happens pretty quick, but it's not widespread. Neuronal is something that is really important to this class, and it's something you've heard about already. This is the equivalent of texting your friend during class, okay? I saw Bill Lowndes texting someone earlier. I was a little offended. So if you could put your phone away, sir, that'd be great. And the key to neuronal transmission is that it's really about electrical movement, okay? That's why it happens so fast, because I've got neurons that span all the way from here to through my spinal cord, or from all the way from my spinal cord out to the tips of my fingers, okay? And that's just electrical transmission via action potentials, which you've learned a lot about. Now, granted, when one neuron talks to the next neuron, it's kind of a relay. It's got to use a neurotransmitter to communicate that, but you can really focus for the purposes of this example on that fast electrical action potential thing, and the fact that it's a lot more specific than what we're about to see, which is endocrine, which is long range. It's all about chemical messengers traveling through the bloodstream. Okay? Hormones in the blood. That's what endocrine signals are about. This is a lot more like Steven sending you guys an email to the entire class, but it takes a long time to get there, right? It's going to take a little bit longer, but it's going to enable us as a class to coordinate our behavior so we all show up to the right place at the right time, wearing the right thing, whatever. Okay, so you guys get that. Questions about those different mechanisms of communication? Okay, so endocrine, clearly the focus of today. Things are traveling through the bloodstream. It's going to take a while to get there, but it's going to allow us to coordinate a lot of different things. Now, what would we want to coordinate? 2 kind of really exciting things that we can coordinate. One is these giant transformations that occur during life. Things like, metamorphosis of, good, I see some mimicking back there. That's very good. I want everyone to do this with me right now. Metamorphosis. Good. So basically, you want all the cells in that organism to kind of change in coordination with each other. Same thing for a couple other things which I'm about to talk about. That's in contrast to, it doesn't have to be an adult organism, it just has to be a functioning organism who is in a certain environment. And in that environment, oh crap, I need to get all my cells on the same page because we've got to address this particular environmental thing. So I'll give you a couple examples. One really great coordinated developmental transformation was the transition from young Harry Potter to Daniel Radcliffe, right? He was in that weird horseplay thing, got a lot of publicity. And you can see, like, a lot of his cells had to change kind of all at the same time, and you get the point. Same thing, I've kind of mentioned these, as opposed to a kind of coordinated response into a specific environmental thing, which might be a stressful situation. We're going to hear about that a little bit later. You want to make sure all your organs and cells are doing the right thing for stress mode or sexual arousal mode, whatever it is. We're going to hear a little bit more about that later. So, Will has the mic.
All right, so that's sort of the background context, putting that stuff into play for today. So some very important things you have to know about hormones overall. We wrote here peptide versus steroid. There's technically a third kind of hormone, which is single amino acid derived hormones. But we're basically going to talk about the distinction between peptide and steroid hormones today. And sort of structural differences and what that means in terms of their transport in the bloodstream, the effect they have on target cells, and then we'll see different things about their duration and other cool things like that. So, structure generally, as you can see here, we put up a picture of insulin in peptide form. We wanted to make it look like a protein. Peptides, of course, and amino acids being the precursors to proteins. We see sort of here, this is a typical peptide hormone made from amino acid precursors, and a key term associated with this is the notion that it's hydrophilic. So this idea that it's water-loving. This plays into the sort of transport mechanisms involved with it and other things which we'll talk about in a moment. So peptide hormones made from amino acids, hydrophilic, water-loving. We'll see some examples. You've heard of some already. Insulin, vasopressin, oxytocin, ACTH, CRH, and more. And then we also have steroid hormones. So these are made from cholesterol precursors. They're hydrophobic, water-hating. Another term we associate with this is lipophilic. Liking of lipids. When we talk about the cellular membrane being made of a phospholipid bilayer, steroid hormones are hydrophobic or lipophilic. They're able to pass through this phospholipid bilayer. Ends up being very important for their mechanism inside target cells. And some examples of this. Classic example, glucocorticoids. We're going to see those all over the place. We already have talked about them a bit. Androgens, estrogen, things like that. Something really interesting about the structure of the hormones. So if we take a steroid hormone, okay, so I'm not going to draw steroid. I'm going to draw a single amino acid, which this actually is looks nothing like a single amino acid hormone, but you sort of bear with me and pretend here. So a lot of these hormones, steroid hormones, single amino acid hormones, peptide hormones, they come from similar precursors, right? In peptide hormones, it's amino acids. In single amino acids, it's often from tyrosine. In steroid hormones, it's from these cholesterol precursors. Why is this interesting? It's interesting because when they come from the same precursor, they end up looking very similar. So one might have this here, so this might be one hormone, and then we might have a nearly identical one underneath, but now it has just another subtle chemical shift. Chemical structure doesn't really matter. The point is that if you're familiar with some organic chemistry, you have just subtle chemical shifts leading to different hormones. Interestingly, from an evolutionary standpoint, this suggests the need for basically having receptors specialized for these subtle chemical differences in the hormones. Norepinephrine and dopamine have just a slight difference like this, right? But the effect, if the receptor wasn't able to distinguish those between those, it would have a drastic effect. So it's very important from an evolutionary standpoint to have receptors that can distinguish these subtle differences in the hormone chemical structure. That sounded really technical. Any questions on that sort of general idea? Is that cool? Okay.
That does remind me of one thing that I didn't make a distinction between when I was talking about neuronal versus hormonal transport. Okay? A lot of times we will kind of throw out these words, and there's a lot of jargony words, norepinephrine, blah, blah, blah. And it's kind of good in your mind to be able to separate between hormones, things that are really these classic things that travel through the blood as signals, and neurotransmitters, right? Neurotransmitters being, they're also chemicals, and they also need to communicate cell to cell, but they are going to be just in that synapse, okay? From one neuron to the next in the synapse that you've already heard about. And just to make your guys' lives awesome, can be overlap. There can be neurons that release their neurotransmitter into the blood, at which point they become hormones. So things like dopamine and epinephrine, right? Things you might associate with neurotransmitters can also be hormones depending on what context and all that stuff. So just something to really focus your attention on when you're distinguishing these signals.
Okay, cool. Thanks. So we talked about the structure very briefly. From that structure, we talked about sort of differences in transport. We talked about hydrophobic versus hydrophilic. The images should have done an animation style pop up. So our peptide hormones are going to be water soluble. They're going to travel freely through the bloodstream. We have, I guess our example here is people riding a roller coaster. These monks are just riding free, looking good. You can imagine the monks as sort of examples of these peptide hormones traveling freely, dissolved through the bloodstream. Not really dissolved, but that's okay. So, peptide hormones traveling freely through the bloodstream. Steroid hormones, however, they're not water soluble, so they need to be bound to a chaperone. This is our example of a chaperone guiding this hormone through the bloodstream. So, it's just sort of an interesting thing to note about their transport. We may talk about this a little later in other contexts. Other interesting things we can learn from the structure. So interaction with the target cell. When Tom was talking about the context here, it's really important that we understand how hormones interact with cells, right? They require a specific receptor for each individual hormone. We talked about the importance sort of the specificity of that. Peptide hormones and steroid hormones sort of have generalized mechanisms that we're going to talk about, ways in which they interact with target cells. Peptide hormones on the left here. So peptide hormones, because they can travel freely through the bloodstream, let me step back. They can travel freely through the bloodstream because of their structural properties. However, those same structural properties, the fact that they're hydrophilic, prevents them from being able to travel through the phospholipid bilayer of a cellular membrane. So the receptors for peptide hormones are on the surface of these cells. Their surface receptor, cellular membrane, the receptors are on the cellular membrane. Because of that, they're typically associated with these what are called secondary messenger responses. The details of this, I think a lot of you guys have been exposed to this in all sorts of biology or human biology, but if you haven't, generally what happens is a peptide hormone binds to a receptor on the cell membrane and then sets off what we always call this cascade response, the secondary messenger cascade response. This is actually really interesting. It can lead to a number of different effects. One thing it can do, which we'll talk about when this is a neuron, is it can activate ion channels, sort of helping ion channels to open or close. We'll talk about that in terms of membrane potential later on. Another thing it can do, and this is the classical example with peptide hormones, is it sets off the secondary messenger cascade that affects proteins within the cell, directly within the cell. So we can think of peptide hormones generally as having action on proteins within the cell. And that's sort of the end effect of the secondary messenger cascade. They also technically can go into the nucleus and affect transcription. However, that we tend to associate more with steroids. And we'll look at some of the complexities and complications of this later on. You'll hear about this. But I would say generally it's important to note that we tend to associate peptide hormones with these secondary messenger cascades affecting proteins existing within the cell at the moment. The onset tends to be relatively quick, and the duration is relatively short. We're going to talk about this in comparison to steroid hormones, and we can see why the relative onset duration is different. So because it's affecting proteins that exist, we can say that the duration is relatively short. And as we said here, may affect protein activity. Steroid hormones, by contrast, they travel through the blood on these chaperone protein carriers. And then when they get to a cell, they're able to diffuse through the membrane and actually bind to receptors located within the cell. Classically, we talk about steroid hormones passing through the membrane, binding to a receptor, and then going directly to the nucleus to affect transcription. So this is sort of our classic example, main effect transcription. the onset then slower and the duration longer. It takes longer to start these transcriptional processes, and it's lasting a lot longer. We're changing the rate of synthesis of proteins rather than changing the proteins that currently exist. Okay. As always, feel free to interrupt if anything seems sort of confusing. So, those are sort of some of the generic examples. Insulin would be a classic example of a peptide hormone. Glucocorticoids is our steroid hormone. We'll see this coming up again and again. We may touch on this again today in the lecture.
Cool. So just to does anyone have questions off the bat? Yes.
For the steroid hormones, it's like a receptor floating in the middle of the cell. Like, would that be like on the nuclear envelope, or is it like on something else?
Great question. So the question was for the receptor for this in this image, by the way, courtesy of Professor Spolsky. Thank you, wherever you are. The receptor is located within. The question is, where is that receptor located for the steroid hormone? Is it on the nuclear membrane? Is it within the cell? Classically, we're going to talk about basically receptors being located in the cytoplasm. There are examples you'll see if you look, even if you did a Google Images search, you might find examples of steroid hormone coming in, binding a receptor, floating in the cytoplasm that's then taken into the nucleus. Basically, we associate the receptors as somewhere within the cell, perhaps on the nuclear membrane, perhaps somewhere else in the cytoplasm, and the steroid hormones bind within there and sort of carry out effects from that.
And you could easily also imagine there being a receptor protein that's already sitting on the DNA, just waiting to be activated, and then when the steroid hormone travels the entire way into the nucleus, that's when it hits the receptor. So yeah, both work. Other questions? Yeah.
What is cholesterol made of? So cholesterol, I have no clue what it's made of. I know generally what its chemical structure looks like. If you look at the steroid hormone picture from the other page, the chemical structure, cholesterol precursors are sort of similar ring structures. You can look it up. I think it's an interesting question. In my mind, what I tend to associate as a relevant question or something that I'm trying to get across more would be that the steroid hormones come from cholesterol precursors, and so I sort of consider that the baseline rather than trying to think about what cholesterol is made from. Interesting question, don't know off the top of my head.
And maybe the most important thing would be that cholesterol is hydrophobic. So if you're going to make stuff from this hydrophobic beginning, it's going to be hydrophobic also. And I just want to make sure people are those are some really jargony terms. Hydrophobic, hydrophilic. There are a lot of synonyms for those terms that are equally jargony. Things like lipophilic and lipophobic, right? Lipo means lipid. So something that is hydrophobic is lipophilic. Does that hurt anyone's brains? Okay. So just kind of maybe spend some time thinking about those words. You can kind of logic through them pretty easily. You might also hear people use the terms polar versus non-polar. Okay, water is polar. So water-loving things, hydrophilic things tend to be polar, and hydrophobic things tend to be non-polar. Don't worry too much about that vocabulary. I just want you to know that there's a lot of different words people might throw around that all essentially are getting at the same thing. Any other questions about this basic setup so far? No. All right. On to chapter 2, which is how does the brain control hormone release? We've established that it'd be a good idea to release hormones in response to certain environmental cues. How can we control that? And as this picture shows, there are many, many different sites in our body where we can release hormones from. We've got the pancreas right there. We've got the testes. We've got all these different glands. Okay, these are just specialized structures that can just pump this hormone into the bloodstream, because that's what hormones are all about. Some that'll come up will definitely be the testes, the ovaries for females. I don't know why they're drawn on the same guy here. Kind of interesting. The pancreas, which is, we keep talking about this thing called insulin. That's released from the pancreas, and it's going to have something to do with food and sugar and things like that. The thymus is another great little endocrine gland. It releases thyroid. No, thymus release. Oh, thymus is where. What endocrine signal does the thymus release? Thymus T cells? I'm not sure those are hormones, though. So anyway, thymus is involved in the immune system. Let's ignore that for now. And blah, blah, blah. You guys get the point. There are many places throughout our body that secrete hormones, and they tend to secrete a certain signal. Now, very excitingly, you'll notice that the brain itself has some endocrine glands in it. Okay, the hypothalamus. We've heard this word hypothalamus a few times. The hypothalamus is an endocrine gland, meaning it can secrete things into blood. The pituitary, right next to the hypothalamus, separated by a little blood, maybe, maybe not. The pituitary also can release hormones into the blood. Okay? And these other kind of lower down peripheral endocrine glands often are regulated by those big master endocrine glands in the brain. Okay? I don't know if you guys have been reading your zebras yet, but I think Professor Sapolsky referred to these as, the brain is the master of these witless organs. Okay? These guys are just kind of like doing whatever the brain tells them to do. And it's the brain that controlling via the hypothalamus and the pituitary is going to be able to control how much testosterone is coming out of those testes. Okay? Does that make sense? The brain controls these peripheral endocrine glands. Okay, so let's zoom in on that hypothalamus pituitary situation. Will really didn't want to include this picture, but I thought it was hilarious, and I can't explain why. Maybe it's the ponytail, I'm not sure. But we're just kind of focusing in on these two different parts. And you'll notice maybe that with something to take away from this picture is simply that the brain is kind of feeding into the hypothalamus, which is feeding into the pituitary. So when we see something, right, that information's processed in our brain, can tell our hypothalamus, oh crap, it's stressful. It can tell the pituitary, and we get this, eventually we can tell the rest of our body what to do via hormones. Awesome. Goodbye, ponytail lady. Okay, here's another just classic Sapolsky image here that we were able to borrow. And this is zooming in once again on just the hypothalamus, and now we're really, really looking at the pituitary. And this is where things are going to kind of hit the fan a little bit. So I want everyone to put your A game on, to mix a metaphor. Okay, so the pituitary. The pituitary are these two kind of things dangling below the hypothalamus. A lot of weird pictures of them. This one's nice because it clearly divides the anterior pituitary from the posterior pituitary. And we're going to make some really, really important distinctions that hopefully you can grasp from this image. And if you can't, let me know. Okay. So first of all, the anterior pituitary secretes hormones. It secretes these things over here. I'll talk about them in a second. The posterior pituitary secretes a different set of hormones. These ones over here. That's one distinction. Another really interesting distinction is how the hypothalamus regulates that secretion. Okay? On the anterior side of things, the hypothalamus, being an endocrine gland, can actually drop some hormones into that little red river of blood right there. Okay? So the hypothalamus is actually releasing a hormone into the blood, and that travels down to the posterior pituitary and tells the anterior pituitary, and tells those cells to then release another hormone into the blood. And once they're in the blood, they can go all the way down to those other target organs we talked about. So that's one system. You've got to actually travel through the blood in the anterior pituitary. The posterior pituitary is actually directly innervated. Okay? So the hormones that are released by the posterior pituitary are actually coming out of those neurons. Okay? And the cell bodies of those neurons are up in the hypothalamus. So, a couple examples. Let's go through the examples. You absolutely do not need to memorize this. We only did this because Will teaches MCAT, and those kids really like their acronyms. They like things that you can say out and put around and put them in bold. So flat PEG is the acronym that people use when they want to memorize a lot of stuff to memorize the anterior pituitary hormones. Some that will be relevant to us that you might want to keep in mind are particularly ACTH. Okay, don't worry about it quite yet, but just kind of plug it in your mind as something cool. And a couple of these other ones might come up as well, but whatever. On the posterior side, we were upset because there was no cool acronym. Is that the right word, acronym? Yeah. For the vasopressin and oxytocin. And we decided that since these are the two hormones that are always getting shouted out in these vole studies, we put some voles on there. And we thought they were cute. They kind of got some Rudolph things going on with the noses. So yeah, whenever you're hearing about vasopressin and oxytocin, those are hormones that are released from the neurons go from the hypothalamus to the posterior pituitary. Now, questions about any of that? That's kind of some neuroanatomy. You're probably not super excited and interested in about that, but it's useful to know. It might come up a little bit. So questions, clarifications? None at all.
There was one.
Oh, yes. Oh my, that's a great question. So I don't know how much we got into kind of, the question was the neurotransmitter / hormones like vasopressin and oxytocin that are released in the posterior pituitary, are those just kind of like sitting there waiting to be released, or do those have to be manufactured and then sent over there? Is that the question?
Yeah, or like if they're released from the neurons in the hypothalamus, right when they need to be released.
Are they released when needed, or are they like created and then sort of sit there?
Okay, so yeah, I would think about this based on your typical understanding of a neuron, which just has a cell body, an axon, and then the axon terminal, which is where it releases neurotransmitter, right? And that axon terminal where it releases neurotransmitter, it's got vesicles. It's basically got these giant balls full of neurotransmitter. Same thing here. You got these giant balls full of oxytocin and vasopressin just waiting to be released.
But aren't those made in the hypothalamus?
Those, like any other neuron, those are synthesized, produced in the cell body of that neuron, which exists in the hypothalamus. And if you're interested in how that works, you got to go on this crazy journey where stuff, proteins, whatever, made in the cell body of that neuron, which you can see up there, has to be carried by these motor proteins down the cytoskeleton. They walk like this. I'm serious. And they have to walk all the way down the cytoskeleton to get there so that they can just wait for the right signal.
That's an excellent question. Also, there's getting at a deeper issue, which is what is it that leads to this hormone release, and we are going to cover that in our chapter 3 coming up.
Yeah. So yeah, clearly there are neurons telling the hypothalamus neurons when to be activated, when to release vasopressin. Other questions about this slide? Is anyone totally confused as to what it is even talking about, or you guys feel okay? All right. One last thing on this one is I'd like to contrast it to Dana's excellent talk on the autonomic nervous system, okay, where you guys learned a lot about a lot of different things, including the fact that the brain can control the autonomic nervous system, right? And the brain actually sends neurons with their cell bodies in the hypothalamus, and instead of sending them to the pituitary, it sends them some other route down your spinal cord and out. And when we were talking about the autonomic nervous system and it being also controlled by the hypothalamus, this is just kind of a reminder, okay? The brain and the hypothalamus are really, really important for regulating a lot of things. In the case of endocrine systems, this is how it works. Cool. Awesome. Now, we're going to look at just one example of kind of zoom back out from the lady with the ponytail to the dude with the ovaries and the testes to remember kind of how all this works together. Okay, and I could have done many different systems, right? Because we had all those different peripheral endocrine glands that needed to leak juice out into the blood. I chose this one because it comes up a lot in the class and it's often referred to. Okay, so it would be the hypothalamic pituitary adrenal axis. The adrenal gland is one of those several glands. Okay? It is regulated by whatever hormones are in its environment. Okay? And whether that hormone is there is thanks to the pituitary and so forth. Okay? There's this kind of cascade of events that allow the brain to control a peripheral gland. And in terms of exactly which hormones do that, we see that the hypothalamus is going to have to leak some CRH into that portal vessel that we saw for the anterior pituitary, right? So those neurons on the upper left would leak some CRH. I might even choose not to tell you what CRH stands for, and let's just say it's CRH. And then CRH travels through that blood until it gets to those neurons where it activates ACTH. Okay? It activates neurons, or sorry, not neurons, it activates endocrine cells to secrete ACTH into the blood, and then that travels through the entire body's bloodstream. Okay, so it's going to go all over the place, but it just so happens that the cells that really like to respond to ACTH are in the adrenal glands. So the adrenal cortex is kind of chilling right on top of your kidneys, having a good time, and when ACTH is present, it will release glucocorticoids. Now that's a big scary word. It's a very important word, okay? Because it's involved. This is the stress hormones. And for those of you guys who have started reading Zebras, you'll have read a lot about glucocorticoids already. They are going to be a really important hormone throughout the second half of the class. So glucocorticoids, stress hormones. One specific one that often gets referenced is called cortisol. Okay, and we don't have to worry about it too much right now. Just remember, stress hormones are getting pumped out. So we kind of understand these first two positive arrows, I hope. Brain sends an arrow to the pituitary, sends an arrow to this via hormones. Now, when cortisol comes out, and it travels through the systemic blood circulation, it's kind of knocking on all the doors of every single cell in the body saying, like, yo, I'm cortisol, I'm here to party. And if that cell has a receptor, this is a theme coming up, then it will respond. Okay? And some of the cells that have receptors for cortisol are in the pituitary, and some are in the hypothalamus. And what that allows for, it allows the pituitary to know how much cortisol is in the system. So when it kind of realizes, oh my God, yay, the ACTH made it there, and cortisol came out, now that I hear the cortisol as the pituitary, I'm going to stop. I'm going to slow down on the ACTH, and I'm going to relax a little bit. And that's something called negative feedback. Okay? Negative feedback, big term in biology and all this stuff, just means that the more cortisol you get it to send out, the less ACTH. Okay? It's kind of a balancing mechanism. Brings us back to baseline so that we stop secreting so much dang cortisol. Same thing exists. There's a whole other area for negative feedback in the hypothalamus. And I think that in the more advanced endocrine lectures, we'll learn about another site of negative feedback, okay? Other parts of the brain that when they hear cortisol can slow this cascade down. So any questions about negative feedback? What is the HPA axis? Why does it represent everything we're learning? Okay.
Okay, great. Thank you, Tom. So Tom pointed out my least favorite slide of the day earlier. This happens to be my favorite slide. This is one of those slides where it looks like it's fairly simple, but actually a lot of thought went into this. So much so that we probably spent like 2 hours with the original slide, scrapped that, and then about an hour ago changed it to this. So here it is. Very important question. Stemming from the things that Tom just talked about, this hormone being released and feeding back to the brain, we're going to investigate that a lot more closely now. We're going to look at hormone action on the brain. We're going to talk a lot about the hormone and receptors for hormones. We mentioned very briefly earlier sort of a generic cellular response to having a hormone bind to a receptor either on the surface or inside the cell. Now we're going to talk about specifically in the brain. So we have hormone to the neuron, an epic journey. The reason this slide took so much work is we were trying to come up with the perfect analogy for a hormone returning to the brain. Something historically that had to go on an epic journey to deliver a message where there were sort of proper receptors to receive the message. So we thought about sort of sticking with the Harry Potter theme and going with Kingsley Shacklebolt's Patronus to Bill LaFleur's wedding. Tom wasn't part of this. This is my idea. But I decided that didn't quite work. We thought about Phidippides, the ancient Greek guy who went from Marathon to Athens to deliver the message that Greeks had defeated the Persians, which is like the route, or why the marathon is 26 miles. Also didn't work. So we stumbled upon this, which is a picture of Paul Revere, the guy who supposedly said the British are coming. The reason this is such a good example, well, or at least we can stretch it to fit here, is that a hormone secreted from an endocrine gland. Let's say we're starting sort of from a target organ here, and Tom just mentioned cortisol coming from the adrenal cortex and going back to the brain. It has to travel a hormone has to travel through the bloodstream. Interestingly, in order for cortisol to be secreted in the first place, it received a signal, right? In this case from ACTH binding to receptors, stimulating the release of cortisol. Paul Revere was sitting out one night. This is the guy back in 1775, actually a week ago today, and like 325 years ago or something like that. 1775, so that math is probably way off, but you can figure it out. He was sitting outside waiting to see these light beacons. He was waiting to receive a signal. And we can think of Paul Revere as sort of a group of hormones here. And when the signal came in, these two light beacons, he set off on this path through the bloodstream, him on horseback through the roads, and with this message. And he didn't actually say the British are coming. He said, boy, what was it? Something like the normals, something like that. I may have written it down. No, I didn't. Okay, well, he said some code word, and basically, this code word, if there were proper receptors for it, they knew how to respond, right? So it wasn't just the British were coming, it was some other word, and if there were receptors for it, they could respond. The relevance in this case is we're talking about going back to the brain. So Paul Revere was going to Lexington, the sort of the central hub, this central nervous system brain, we can sort of stretch it that way, and has to find the proper receptors there to initiate this proper response. So, I don't know, I just like that analogy. So, what we're going to talk about is hormone back to the neuron has to get through the blood-brain barrier. Does the neurons, it's going through the neuron or the group of neurons, do they have the proper receptors for this hormone signal? And once it binds, how does it influence activity in the brain? What is going on there? Questions so far? Did I confuse significantly with that? Okay, cool. So, we're going to move on. Starting with the blood-brain barrier. So the blood-brain barrier. On the left, we have this picture of just blood vessels in the brain. This is here because Tom sort of has a personal mission. He and I both have been confused about what the blood-brain barrier is, what it looks like. He used to envision, you can tell.
It's just really embarrassing, so I'd rather say it myself. I used to think that like the brain was in this barrier that protected it from like the evil blood supply, when it obviously can't be like that because the blood has to get inside the brain and tell every single cell in the brain what to do. And I'm confessing this to you today, that you actually have to picture the blood vessels' access to the brain in a much more complicated way. So the barrier itself is much more complicated as well.
Right, so the barrier is constructed by basically these epithelial cells lining the blood vessels, right? The blood vessels extend throughout the brain, just sort of an image of it there, and we have this blood-brain barrier created by these tight junctions and epithelial cells, sort of a bunch of details that aren't necessarily super important. However, it's interesting because this barrier, this blood-brain barrier, basically tightly regulates what type of things can go inside and outside the bloodstream in the brain. It's very important that we have a lot of control over what's going on in terms of our blood supply in the brain. So hormones, the question comes up, can hormones pass through the blood-brain barrier? We did a bunch of research on this, and basically because these are epithelial cells, phospholipid bilayer, same story. Steroid hormones are lipophilic, they can pass through this phospholipid bilayer, not much of a problem there. Peptide hormones, generally we know that they can't pass through just on their own, but we looked this up. There doesn't seem to really be a problem with peptide hormones getting to the places they need to get in the brain because they have these carrier transports. They have the ability to get through the blood-brain barrier when they need to. So just sort of an interesting point there. First step, getting through blood-brain barrier, it's helpful to know it exists. It might come up in other contexts later on. Things like alcohol. Alcohol can go right through the blood-brain barrier, and that has certain effects on behavior. It might come up later on. Here's this blood-brain barrier. What can get through it? What can't? What does that mean? What are the implications for that in terms of behavior? Something just to sort of know about generally. So, next step, receptors. So hormones, when they're traveling back to the brain, there have to be appropriate receptors for these hormones. Tom talked about cortisol, an example of a glucocorticoid. Glucocorticoid is a steroid hormone. This is an image of a rat brain and glucocorticoid receptors in the rat brain. We also have mineralocorticoid receptors, which we're not really going to focus on in blue. An example would be like aldosterone. For my MCAT people, you know what that is, but we'll, thanks for the nod, Nate. But we're not going to focus on that. So, glucocorticoid receptors. As you can see, there are glucocorticoid receptors throughout the rat brain. Interestingly, however, we see them sort of clumped in certain areas. What's going on there? This might suggest a particular area that's particularly sensitive to these glucocorticoid hormones, right? So these glucocorticoid hormones come into the brain, diffuse through the membrane, and bind to these receptors located in the brain. We see here that one area is the hypothalamus. Tom already mentioned one effect of what's going on there, potentially sort of these negative feedback mechanisms, right? Glucocorticoids coming back and helping to regulate or downregulate the release, maybe I shouldn't say downregulate, but basically suppress the release of CRH and eventually ACTH and other things affecting cortisol's release in the first place. We also see these receptors clumped in the hippocampus here. So another interesting potential other type of negative feedback thing going on there. Stay tuned. We may hear more about this later on. Interesting point, though, is that for every single hormone, we're going to have unique receptors throughout the body, but we're looking at the brain right now. So we're going to have unique receptors for these hormones in the brain. We're going to want to be paying attention to when we're looking at behaviors. We're going to want to think about basically where are these receptors in the brain, what types of receptors are they, that sort of thing. On this slide we wrote receptors, location, type, and how many. So I briefly just talked about location, the location of receptors, how that can matter. Type of receptors. You can have within an individual, you can have different types of dopamine receptors, for example, right? You can have multiple different types of receptors for a particular hormone, and this can lead to different effects depending on what type this is. We also heard about this on sort of the populational level, right? A variation in the type of vasopressin receptor and how that can lead to sort of monogamous versus tournament species type behavior. I guess maybe stay tuned for this, but we've also heard about these things before. So types of receptors are interesting, location of receptors, how many receptors? This is another question that becomes very important. If we have a lot of receptors, we can basically predict that there's going to be a higher sensitivity to the hormone in that area. The low amount of receptors, not as sensitive. When we look at when we talk about Menie's rats, epigenetic mechanisms, this is supposed to be basically affecting the expression of receptors in certain parts of the brain, and then we can see the behavioral impacts of that. So another thing briefly related to how many receptors we have the level of hormone and we have the level of receptors. And both of these things basically impact the behavioral output. An interesting effect we can begin to see happening is that the level of hormone can affect the upregulation or downregulation of receptors. You might start out with a certain amount of receptors, and then if you're flooded with tons and tons of hormone, these receptors then might start to downregulate. The cell might sort of downregulate the amount of receptors expressed on the surface of the cell, and you might see downregulation. Conversely, we might see upregulation if it's not receiving enough of this hormone. Stay tuned for more of this, complexity with this, complications with this, etc. Any questions so far on this stuff? Okay, cool. So, hormone action on the brain. Next, we turn to basically looking specifically at a neuron. As I mentioned before, we talked about sort of the effects on cells. We talked about peptide hormones and the secondary messenger cascades. We put the image back up, talked about steroid hormones and generally affecting transcription. Now we basically want to zoom in on a neuron and see what types of effects could be happening on these cells, these brain cells. So we wrote down here, we summarized the very nicely, potential effects of hormones on neurons. Once the hormone binds to a receptor, we can have it change the membrane potential via ion channels. So maybe secondary messenger cascade, if basically some molecule of some sort bumps into another channel, opens it more. Now you've changed the membrane potential of the cell. It either becomes easier or more difficult to start an action potential, something like that. We can change transcription of genes. So it can go in, like we saw this with the steroid hormone going in and basically binding to the DNA, affecting transcription of genes. In this case, the genes, they could be receptors. This might be sort of a mechanism for upregulation or downregulation of receptors, something like that. We can also have, of course, change in protein activity and transport. This, of course, is crucial, right? Proteins could be involved with tons of things. Neurotransmission, formation of new synapses, the things listed there. Basically, hormones can have impacts on all of these different areas. Protein activity, transcription, membrane potential, all these different things. And so at an individual cellular level, we can begin to see sort of the wide array of impacts of hormone binding, what that can do. If you step back and think about this as sort of networks of neurons, not just individual neurons, but networks of neurons, now you can begin to see how hormones can start to shape behaviors more overall. Just these networks of neurons, and those behavioral outputs being sort of the output of these networks. Yeah, question?
When you talk about impact on neurons, do you mean cells that are in the brain and the central nervous system, or are you just talking about the brain?
Great, so question, actually, can you repeat that for me?
Are we talking whole central nervous system, or just the brain?
Whole central nervous system or just the brain? So in this case, I'm talking about neurons that are in the brain, but these might project anywhere. As Tom said earlier, neurons can project to other areas within the brain, can project down the spinal cord and elsewhere. So the impact could be essentially anywhere that these neurons could project when we're talking about the effect of hormone input on neurons in the brain. The key is the neurons, right, where they project, they'll have a certain impact on, they'll have some certain output wherever they project to. But in the first place, they have to have a receptor for this hormone to bind. So it's just something interesting to note. Other questions? That's not a question. Okay, cool. Good, thanks. So I think that's kind of summarizes my thing on that. Tom, did you have anything to add to that?
I'd say the more interesting things that when we're talking about neurons changing activity, neurons in the brain is where the interesting stuff is happening. Maybe the lordosis reflex has more of the, how hormones might influence how likely you are to do your little back arch raise, but who knows?
Okay, cool. So, so we talked about in the nervous system lectures how behavior is affected by the nervous system. Now we've talked about how hormones can affect the nervous system and therefore sort of affect behavior as an output there. We're going to give you a little preview of some areas we might see hormones interacting in future lectures. Stress. We're definitely going to see glucocorticoids associated with stress response. Here's Harry and Sirius looking sort of stressed. Sexual behavior. Testosterone, estrogen, vasopressin, oxytocin. We're going to see these things as players in sexual behavior lectures. Aggression, testosterone again, glucocorticoids, estrogen, epinephrine. Depression. Okay, there's something about that picture. All right. In depression, depression, glucocorticoids, thyroid hormone, estrogen, progesterone, melatonin. Great. So some things.
Yeah, just something to add for this slide is we're kind of throwing a bunch of random hormones at you that you might not be familiar with yet. Something to think about is how every single thing we've told you thus far regarding receptors and hormone levels and room for individual variation and all these things, levels, where they are, what type of receptors might influence these things. That's going to be kind of how we walk through a lot of these behaviors.
Cool.
And then I think the last slide is just take-home points, things that are really excellent for you to try to walk away with, so don't walk away if you don't understand most of these things. But steroid versus peptide hormones and the importance of what it means to be hydrophobic. Okay? Pretty much a lot of the consequences for how we distinguish between these two things depend on whether you're hydrophobic or hydrophilic, so it makes it pretty easy to logic through if you know that basic thing, whether you need the chaperone or not, and where your receptor is going to be. The nervous system control of hormone release. You guys, I kind of hit that into the ground, the HPA axis as one example of how the brain could control peripheral endocrine glands. And then finally, when those hormones are released, how can they influence behavior via neurons? Kind of the long trials and tribulations of Paul Revere. A really excellent analogy, I think. I think you guys would all agree. Basically that's it. So if you guys have any questions, you can shout them out or just come down here and ask us because you guys have fun things to do today. Thank you.
For more, please visit us at stanford.edu.